We're trying to learn about the genetic determinants of kidney function in minority groups. As we are all aware, the progression of kidney disease to ESRD is uh, fourfold higher in the African American population, and it also affects the Latino population at a higher rate. So we wanted to learn um, genetic variants in addition to APOL1 that played a role in faster progression to end-stage renal disease so that we could complement the information that we get from using just APOL1 and actually sickle cell trait. So we took advantage of the Million program, which is the largest biobank in the world currently. And we use a sample size of about 82,000 African Americans. The genetic analysis per se is a standard approach. And we just use the linear additive model to a cross-sectional approach of kidney function, or EGFR, that was measured at enrollment. We were able to adjust for BMI, age, gender, and sex, and 10 principal components of ancestry. So what really is great about these analysis is the sample size. And we're hoping that in the future, we can have even further participants in the minorities analysis. Also, because this is linked to the electronic health record, we have the opportunity, which we are not presenting in this current presentation, of doing many post-GWAS analysis, such as GWAS, and genetically predicted gene expression, which help us to bring all those associations that we see in the GWAS to a more translational context and be able to inform a little more of the implication of the different variants that we are getting from the genome-wide association analysis. So in addition to the African American Genome-wide Association studies, you will learn that in this particular study, or in this particular meeting, we are presenting complementary analysis, which include an APOL1 interaction by Cassian Robinson, and we're also doing an analysis in a GWAS for Hispanics. So all of these different Genome-wide Association studies do complement each other, and help to bring further that very particular interest of us, which is just learning what other genetic variants are unique to minority groups and have impact in their higher risk of progressing to end-stage renal disease.
We also had, like, during the GWAS, we used different methods. Like, our statistician brought up something called effective sample size, which was just a nice way to have nicer QQ plot results, but also to generate more results. So we were able to capture the sickle cell trait. We were able to actually get some of these rarer variants in there via that method as well. And then we also were able to use GCTA Kojo to actually do independent hits from our analyses. So that actually helped. And that's what will be presented are the independent hits. Well, for the ones that were already identified, replication is a huge deal. So we do need to make sure. So of course, seeing APOL1 pop up again and GADM and some of these other, or sickle cell trait, that's always a positive sign too, because that means that we know these aren't spurious associations. These are things that are already identified in the literature. And the novel loci, those have a lot of them have like different cellular level effects. So being able to sit down and kind of they give new breath to try to study out more, and some of them aren't seen in kidney disease as much. Some of them are seen more in like cardiovascular issues. So it's just an interesting way to explore the field more and kind of see how things link up.
Yeah, that's a great question regarding the three novel loci. One of them we known for being associated with accelerated atherosclerosis, and that is the BCA1. That's an important link, as we know that patients with chronic kidney disease have an accelerated atherosclerosis process that we're still trying to figure out the mechanisms and pathways to see if we can change that. For the other two loci, we're not sure right now of the mechanisms that link them to chronic kidney disease. However, as I mentioned before, we're doing many post-translational analysis, including the genetically predicted gene expression in kidney tissue. We also are linking them to genome-wide association analysis, and we're using them against other drug relationship databases. So these type of approaches are going to help us to figure out these other two loci, where are their, how are they related to gene expression in the entire body and in the kidney, so that we can understand further their mechanisms. And so I guess that in the near future, after all these post-GWAS analysis, we will be able to inform the community about the potential implications that they have in the gene expression of other kidneys, or if they have functional implications on their own.